Topic: Frequency distributions and their descriptive statistics in ecology and evolution. Recall. Topic: Frequency distributions and their descriptive statistics in ecology and evolution. Recall. A frequency distribution is a representation of how often different values or ranges of values occur within a dataset. In the context of ecology and evolution, frequency distributions are often used to describe the distribution of traits within populations, the frequency of species occurrences in different habitats, or the distribution of environmental variables like temperature or rainfall. Key elements of frequency distributions include classes or intervals. The range of data is divided into intervals or bins and the number of data points in each interval is counted. Frequencies, the number of occurrences of a particular data value or range of values within each class. Relative frequencies, the proportion of the total number of data points that fall into a particular class. A common example in ecology might be the distribution of tree heights in a forest, while in evolutionary biology, it could be the distribution of a particular genetic trait across different populations. B. Comprehension. In ecology and evolution, the analysis of frequency distributions helps to understand patterns within populations and ecosystems. For example, frequency distribution of species in a community may reveal whether a few species dominate, or whether there is a relatively equal distribution of species, e.g., normal, or log normal, distribution. Trait distribution. The frequency distribution of a particular trait, e.g., beak size in birds within a population can indicate whether selection is acting on that trait. Descriptive statistics are used to summarize the central tendency, dispersion, and shape of the data. Descriptive statistics are key to understanding the patterns of variation in ecological data or evolutionary traits. C. Application. Frequency distributions and descriptive statistics are applied in numerous ways in ecology and evolution. Ecological communities. In community ecology, the frequent distribution of species abundance often follows patterns like the log normal distribution, which is observed when a few species are abundant and many species are rare. Analyzing this distribution helps ecologists understand biodiversity and species dominance in ecosystems. Example. In a tropical forest, a frequency distribution of tree heights may show that most trees are of medium height, with few being very tall or very short. This can indicate the competitive structure of the forest and how trees have adapted to the environment. Population genetics. Descriptive statistics are often used to summarize genetic variation within a population. Measured allele frequencies and the distribution of genotypes provide insights into the evolutionary processes at play. For example, the frequency distribution of alleles for a particular trait can help understand the impact of natural selection, genetic drift, or gene flow. Example. Population of a species. Frequency distribution of a genetic mutation might indicate whether the mutation is neutral, advantageous, or deleterious. Trait selection and evolution. In evolutionary biology, the frequency distribution of traits in a population over time can show how traits are being selected for or against. For example, the distribution of body size or beak shape in a bird population can indicate how these traits are influenced by environmental factors or natural selection. Example. In a population of finches, the frequency distribution of beak size might change over generations due to changes in available food sources. Descriptive statistics such as the mean and standard deviation of beak size will provide insight into how selection pressures are shaping the trait distribution. Biodiversity studies. Descriptive statistics are also used to assess biodiversity within ecosystems. Species richness and evenness can be calculated from frequency distributions of species abundances, providing important metrics for conservation biology and ecosystem health. Example. A frequency distribution of species in a grassland could be analyzed to determine the biodiversity of the habitat. If the distribution is highly skewed, with one species dominating, this might indicate a loss of biodiversity or ecosystem imbalance. D. Analysis and synthesis. By analyzing frequency distributions, ecologists and evolutionary biologists can synthesize important information about populations and ecosystems. For example, variation in normal distribution. Ecological and evolutionary traits, such as body size or genetic variation, often follow a normal distribution, where most individuals cluster around a central value, the mean, and fewer individuals have extreme values. This allows for the application of various statistical techniques, including hypothesis testing and analysis of variance ANOVA to compare group skewness and kurtosis. The shape of a frequency distribution can provide insights into ecological or evolutionary processes. For instance, a positively skewed distribution might indicate a population where most individuals have low values for a particular trait, e.g., short beaks, but a few individuals have very high values, e.g., long beaks. The kurtosis of the distribution can also indicate the presence of extreme values, outliers, which may suggest evolutionary outliers or rare ecological events. Outliers in evolutionary fitness. Outliers in a frequency distribution might indicate individuals with exceptional traits, which may confer evolutionary advantage. For instance, an individual with an exceptionally large beak in a population of birds might have a higher fitness if it can access a unique food resource that others cannot. The interquartile range, IQR, and standard deviation are used to identify such outliers. Comparing populations. Comparing frequency distributions of the same trait across different populations can help identify differences in ecological conditions or evolutionary pressures. Descriptive statistics such as mean, variance, and standard deviation can help determine how two populations differ and whether these differences are statistically significant. Descriptive statistics and frequency distributions. Descriptive statistics provide summary measures that help interpret the frequency distribution and understand the central tendency, spread, and shape of the data. Common descriptive statistics include mean, average. The mean is the sum of all values divided by the number of observations. It represents the center of the distribution. Formula. Mean equals n area summation I equals 1 nxi n mean equals n. N area summation I equals 1 nxi. Median. The median is the middle value when the data are arranged in ascending order. It is less sensitive to outliers than the mode. The mode is the value that occurs most frequently in the data. Range. The range is the difference between the maximum and minimum values in the dataset. Formula. Range equals max minus min. Range equals max minus min variance. The variance measures the spread of the data points around the mean. It is calculated by averaging the square difference from the mean. Formula. Variance equals n area summation I equals 1 n xi minus mu 2n. Variance equals n. N area summation I equals 1 n xi minus mu 2. Standard deviation. The standard deviation is the square root of the variance and represents the average distance of data points from the mean. Formula. Standard deviation equals variance. Standard deviation equals variance. Skewness. Skewness measures the asymmetry of the data. Optative skew indicates a longer right tail, while a negative skew indicates a longer left tail. Kurtosis. Kurtosis measures the tailedness of the distribution. High kurtosis indicates more outliers, while low kurtosis indicates fewer outliers. Practice questions previous GATE papers. Q1. The heights of trees are measured, and the following data in cm is obtained: 140, 152, 153, 160, 162, 165, 170, 170, 175, 180. Calculate the mean, variance, and standard deviation for this data. Answer. Mean. Mean equals 140 plus 152 plus 153 plus 160 plus 162 plus 165 plus 170 plus 170 plus 175 plus 180 10 equals 161.7 mean equals 10. 140 plus 152 plus 153 plus 160 plus 162 plus 165 plus 170 plus 170 plus 175 plus 180 equals 161.7 variance. Variance equals 140 minus 161.7. 2 plus 152 minus 161.7. 2 plus 180 minus 161.7. 210. Variance equals 10. In an study, the number of individuals in several populations of a species is recorded: 12, 15, 18, 22, 25, 30, 35, 36, 40, 45. In an ecological study, the number of individuals in several populations of a species is recorded: 12, 15, 18, 22, 25, 30, 35, 36, 40, 45. Calculate the mode and range of the data. Answer. Mode. There is no mode since all values occur only once. Range. Range equals 45 minus 12 equals 33. Range equals 45 minus 12 equals 33Q3. Given the following frequency distribution of bird sizes in cm in a population, calculate the mean size. Size cm frequency 10.3. 12514816418 answer. First, multiply each size by frequency. 10 times 3 plus 12 times 5 plus 14 times 8 plus 16 times 4 plus 18 times 2 equals 30 plus 60 plus 112 plus 64 plus 36 equals 302. 10 times 3 plus 12 times 5 plus 14 times 8 plus 16 times 4 plus 18 times 2 equals 30 plus 60 plus 112 plus 64 plus 36 equals 302. Total frequency equals 3 plus 5 plus 8 plus 4 plus 2 equals 22. 3 plus 5 plus 8 plus 4 plus 2 equals 22. Mean equals 302. 22 approximately equals 13.73. cm 22,302. Approximately equals 13.73 centimeters. Understanding frequency distributions and applying descriptive statistics is essential in interpreting ecological data and evolutionary patterns. By using these techniques, researchers can derive meaningful insights into population dynamics, genetic variation, and ecological interactions. You said, mean. ChatGPT said, ChatGPT. Topic, mean in ecology and evolution, recall. The mean, also known as the average, is a measure of central tendency that all the values in a dataset and divides by the number of values. It is one of the most commonly used statistical measures to summarize data, as it represents a typical value in the dataset. In ecology and evolutionary biology, the mean is frequently used to describe the central value of various biological measurements, such as body size, beak size, population densities, or genetic trait frequencies. Formula for mean. Mean equals n area summation I equals 1 nxi n mean equals n. N area summation I equals 1 nxi. Where n area summation I equals 1 nxi n area summation I equals 1 nxi is the sum of all the data points. N. N is the total number of data points. For example, in a population of organisms, the mean might be used to describe the average size of individuals, the average number of offspring produced, or the average genetic distance between individuals in a population. B. Comprehension. In the context of ecology and evolution, the mean is an essential measure for understanding the typical values within a dataset. It helps provide a baseline against which other data points can be compared. However, it is important to note that the mean can be influenced by outliers, values that are much larger or smaller than the rest, which may skew the result. Ecology. In an ecological study, if researchers are measuring the height of trees in a forest, the mean would give the average height of all the trees. However, if there are a few extremely tall trees, the mean might be higher than expected. Evolution. In an evolutionary context, the mean can represent the average value of a trait, e.g., average beak size in a bird population. Insight into the typical expression of that trait in a population, which is important when studying natural selection, genetic drift, and adaptive radiation. C. Application. The mean is applied in various areas of ecological and evolutionary research to summarize complex data. Here are a few specific applications. Population ecology. In population ecology, the mean can be used to calculate the average population size over a period of time or the average species density within a certain area. For example, if a researcher wants to estimate the average number of individuals of a species in different habitats, the mean number of individuals per habitat can be calculated to determine habitat preference. Example. A study on tree density in a forest reports the following data for 3 regions. Region 1 50 trees per hectare. Region 40 trees per hectare. Region 3 60 trees per hectare. The mean tree density across the 3 regions would be mean equals 50 plus 40 plus 63 equals 50. Trees per hectare. Mean equals 3. 50 plus 40 plus 60 equals 50 trees per hectare. Evolutionary biology. In evolutionary studies, the mean is often used to describe the average trait value of a population, such as body size, beak size, or genetic variation. It allows researchers to track how populations change over time in response to selective pressures, such as predation or resource availability. Example. If researchers are studying the beak size of birds in a population before and after a drought, the mean beak size can show whether selection has favored individuals with larger or smaller beaks due to changes in available food resources. Genetic variation. In evolutionary genetics, the mean can describe the average allele frequency in a population. It helps in understanding the genetic diversity and evolutionary forces acting on populations. A shift in the mean allele frequency might indicate natural selection or gene flow. Conservation biology. The mean is also useful for summarizing population health and biodiversity metrics in conservation biology. For instance, researchers use the mean to summarize the average genetic diversity of individuals within a population, which could help assess its susceptibility to inbreeding depression or its ability to adapt to environmental changes. D. Analysis and synthesis. While the mean is a useful summary statistic, it has limitations, especially in skewed distributions or when there are outliers. Here's a deeper analysis. Influence of outliers. The mean can be heavily influenced by outliers or extreme values. For example, one individual in a population of birds has a significantly larger beak than the others. This extreme value can raise the mean beak size, even though most individuals may have similar beak sizes. Even with median and mode. In datasets where outliers or distributions are present, the median, the middle value of the dataset, or the mode, the most frequent value, might be more appropriate measures of central tendency than the mean. For example, in a dataset of species population sizes, the mean might not represent the typical population size if most populations are small but a few are very large. Changing mean over time. In evolutionary studies, tracking changes in the mean of a trait, e.g., body size or genetic frequency, over time can reveal insights into evolutionary processes. For example, if the mean body size in a population of organisms increases over several generations, it could indicate the action of directional selection. Mean and evolutionary forces. The mean can also reflect the balance between different evolutionary forces. For example, in a population undergoing stabilizing selection, the mean trait value will remain relatively constant over time. In contrast, in a population experiencing disruptive selection, the mean trait value may shift or split into two distinct groups. Practice questions. Previous gate papers. Q1. The following are the weights kilogram of 5 different species of birds: 1.8, 2.4, 2.9, 3.2, 2.7. Calculate the mean weight of the birds. Answer. Mean equals 1.8 plus 2.4 plus 2.9 plus 3.2 plus 2.75 equals 13. 5 equals 2.6. Kilogram mean equals 5. 1 point plus 2.4 plus 2.9 plus 3.2 plus 2.7 equals 513 equals 2.6 kilograms. Q2. A study is conducted on the number of offspring produced by a population of mammals. The following offspring numbers are recorded for 6 individuals: 4, 6, 5, 7, 8, 5. Calculate the mean number of offspring. Answer. Mean equals 4 plus 6 plus 5 plus 7 plus 8 plus 5 6 equals 35. Understanding the mean and its application in ecology and evolution helps researchers summarize data effectively and identify trends or patterns in populations. The measure is foundational in many areas of ecological research and evolutionary analysis. Who said? Variance. ChatGPT said. ChatGPT. Topic. Variance in ecology and evolution. Recall. Variance is a statistical measure of the spread or dispersion of a dataset. It quantifies how far each data point in a set is from the mean average of the dataset. The higher the variance, the more spread out the data points are. The lower the variance, the more concentrated the data points are around the mean. Formula for variance. For a population. Variance. Sigma 2 equals n area summation I equals 1 n xi minus mu 2 n. Variance sigma 2 equals n area summation I equals 1 n xi minus mu 2, where xi represents each data point in the dataset. Mu is the mean of the dataset. N is the number of data points for a sample instead of the entire population. Sample variance. S2 equals n area summation I equals 1 n xi minus x 2 n minus 1. Sample variance S2 equals n minus 1 n area summation I equals 1 n xi minus x 2, where x is the sample mean. Variance is measured in the square of the units of the original data. For instance, if the data is in kilograms, variance will be in kilograms squared. B. Comprehension. Variance helps in understanding how much individual data points differ from the mean. In ecological and evolutionary studies, it gives insight into the diversity or variability within populations, helping researchers assess whether a population is homogeneous or diverse. Ecology. Ecological research. Variance can be used to describe the variation in attributes like species population sizes, body mass, or resource availability. For example, the variance in the number of individuals in different patches of habitat can indicate how evenly resources are distributed across the landscape. Evolutionary biology. In evolutionary biology, variance plays a critical role in genetic diversity. Patterns in traits such as beak size, fur color, or flower shape within a population can suggest adaptive radiation or evolutionary pressures. Small variance in a trait might indicate stabilizing selection, where natural selection favors the average phenotype. C. Application. Variance is widely applied in both ecological and evolutionary studies to assess the distribution of data and understand how populations or traits evolve over time. Population ecology. In population ecology, variance can be used to measure the variation in population densities in different areas. High variance in the population size across different regions might indicate that certain areas are more conducive to supporting large populations, while others may be less favorable. Example. If a study of a plant species records the following data on the number of individuals per hectare across 5 regions, 150, 200, 250, 300, 500. The variance will give an idea of how much the population size varies between the regions, highlighting areas with significantly higher or lower densities. Evolutionary studies. Variance is essential in understanding trait variation within a population. For example, size in birds. The variance helps in assessing whether there is a broad range of sizes, which might suggest ongoing evolutionary processes like disruptive selection, or if the population is more uniform, which might suggest stabilizing selection. Example. A researcher might measure the beak length in a bird population across multiple generations. If the variance in beak length is high, it could indicate that directional selection is occurring, as the population evolves towards a new average trait value. Genetic variation. In evolutionary genetics, variance is used to measure the variation in allele frequencies within a population. A high variance in allele frequency suggests a genetically diverse population, which may be more adaptable to environmental changes. Low variance may indicate genetic bottlenecks or reduced diversity, which can make a population more vulnerable to extinction. Conservation biology. Variance in traits, such as genetic variation or reproductive success, is crucial in assessing the health of a population. A population-wide variance in reproductive success may indicate that some individuals are more fit and contribute more genes to the next generation, whereas a population with low variance may suggest that the entire population is struggling. D. Analysis and synthesis. Variance is a critical measure in both the analysis and synthesis of ecological and evolutionary data. While it helps quantify diversity and variability, it is important to understand its limitations. Influence of outliers. Variance is sensitive to extreme values or outliers. A single data point far from the mean can disproportionately increase the variance, making it appear that the data is more spread out than it actually is. For example, in a study of animal body sizes, if one individual is much larger than the rest, it can skew the variance, making it seem as though there is more variation in the population than is representative of most individuals. Standard deviation. The standard deviation, which is the square root of the variance, is often more useful because it has the same units as the original data, making it easier to interpret. For example, if the variance in body mass is 25 kg squared, the standard deviation will be 5 kg, which is easier to interpret in the context of the dataset. Comparing populations. Variance allows for the comparison of different populations. For instance, a population of birds with high variance in beak size might be undergoing divergent evolution, whereas a population with low variance might be undergoing stabilizing selection. Evolutionary processes. Variance is often used to infer evolutionary processes. A decrease in variance over generations could indicate stabilizing selection, where the population is becoming more uniform. In contrast, an increase in variance might suggest the action of disruptive selection, where extreme phenotypes are favored. Practice questions. Previous gate papers. Q1. Given the following data representing the number of offspring produced by 6 individuals of a species, 3, 4, 6, 8, 10, 12. Calculate the variance. Answer. Step 1. Find the mean. Mean equals 3 plus 4 plus 6 plus 8 plus 10 plus 12. 6 equals 43. 6 approximately equals 7.17. Mean equals 6. 3 plus 4 plus 6 plus 8 plus 10 plus 12 equals 643. Approximately equals 7.17. Step 2. Calculate the squared differences from the mean. 3 minus 7.17. 2 equals 17.99. 4 minus 7.17. 2 equals 10.03. 6 minus 7.17. 2 equals 37. 3 minus 7.172 equals 17.99. 4 minus 7.172 equals 10.03. 6 minus 7.172 equals 1.3. Variance is a key measure of the variability of traits or populations, crucial for understanding evolutionary processes and ecological patterns. By calculating and analyzing variance, researchers gain insights into diversity, adaptation, and selection within populations. You said. Coefficient of variation. ChatGPT said. ChatGPT. Topic. Coefficient of variation, CV, in ecology and evolution. Recall. The coefficient of variation, CV, is a statistical measure of the relative variability in a dataset. It is defined as the ratio of the standard deviation to the mean, expressed as a percentage. The CV allows for comparison of variability between datasets with different units or scales, making it particularly useful when comparing datasets with different means or when comparing variability across populations in ecological and evolutionary studies. Formula for coefficient of variation. C equals sigma mu times 100. CV equals mu. Sigma times 100 where sigma. Sigma is the standard deviation of the dataset, mu. Mu is the mean of the dataset. The CV is a dimensionless quantity, meaning it does not have units, making it ideal for comparing the variability of datasets with different units, e.g., comparing the variation in body size of 2 species measured in different units. B. Comprehension. The coefficient of variation, CV, helps to quantify the relative variability within a dataset. It is particularly useful when comparing datasets that are on different scales or have different means. The CV expresses how large the standard deviation is relative to the mean, providing a normalized measure of variability. In ecological and evolutionary studies, a high CV indicates high variability relative to the mean, which may suggest significant diversity or instability in the system. A low CV suggests low variability relative to the mean, indicating a more stable or homogeneous system. Ecology. In ecology, the CV can be used to compare the variability in population densities across different habitats. For example, comparing the CV of tree heights in a dense forest and a sparse woodland can help determine which habitat has more consistent tree growth. A higher CV in one area would suggest more variability, which could be due to environmental factors like soil quality, light availability, or moisture levels. Evolutionary biology. Evolutionary biology, the CV can be used to assess phenotypic variation within a population. For example, the CV of beak size in a bird population can help indicate how much diversity exists within the population. High CV in a trait may suggest a population that is experiencing divergent selection, whereas low CV may indicate stabilizing selection that favors average traits. C. Application. Coefficient of variation is widely used in ecological and evolutionary studies to assess the variability of traits, populations, or environmental factors. It helps compare the relative consistency of data across different populations, environments, or species. Ecological studies. C can be used to evaluate resource distribution in ecosystems. For instance, if you measure the leaf area of plants across different environmental gradients, the CV can highlight areas where plants are experiencing more variability in growth, which might be a response to resource scarcity or environmental stress. Example. If researchers are comparing population size of a species in two different habitats, and find that one habitat has a CV of 25% and the other has a CV of 50%, it would indicate that the second habitat has more variability in population size. This could be due to factors such as resource fluctuations, predator presence, or habitat disturbance. Evolutionary studies. In evolutionary biology, the CV is often used to study the variation of traits like body size, reproductive success, or genetic diversity across different populations. A higher CV may suggest a population undergoing directional selection or experiencing environmental heterogeneity. A lower CV may indicate a more uniform population that is under stabilizing selection. Example. In a study of flower color variation in a population of plants, a researcher may find that the CV of flower color is higher in regions with variable soil types, suggesting that environmental factors are influencing the diversity of flower colors in the population. Conservation biology. The CV is used to assess the genetic variation within a species. High CV and allele frequencies may indicate high genetic diversity, which is important for the long-term survival and adaptability of the species. Simply, low CV might signal genetic bottlenecks or inbreeding risks, which could threaten the population's viability. Example. Conservation biologists studying a rare species might calculate the CV of reproductive success among individuals in different populations. A high CV might suggest that some individuals are much more successful at reproducing than others, potentially driving genetic divergence between populations. D. Analysis and synthesis. The coefficient of variation, CV, is a powerful tool for analyzing and synthesizing data in both ecological and evolutionary research. By comparing CVs across different datasets, researchers can assess the relative variability in populations, traits, or environments, evaluate stability versus instability in systems, identify potential areas of adaptation, selection, or stress. However, there are some considerations and limitations when using CV. Dependency on mean. The CV is sensitive to the mean of the dataset. If the mean is very small or close to zero, the CV can become misleading. For example, if the mean size of a population is very small, a small variation might result in a very high CV, even though the variation is not substantial in absolute terms. Interpretation of CV. A high CV does not always indicate a problematic or unsustainable system. In evolutionary contexts, a high CV could signify that a population is experiencing adaptive radiation or divergent evolution, which can lead to the development of new species or traits over time. Comparing across datasets. The CV is especially useful when comparing variability between datasets with different scales or units. For instance, when comparing the growth rates of two plant species, one measured in cm per day and the other in inch per day, the CV can help compare the relative variability in their growth patterns, even though the units are different. Practice questions. Previous gate papers. Q1. A population of birds has the following body weights in grams: 2.5, 3.0, 3.2, 3.5, 3.8, 4.0, 4.2, 4.5. Calculate the coefficient of variation. Answer. Step 1. Calculate the mean. Mean equals 2.5 plus 3.0 plus 3.2 plus 3.5 plus 3.8 plus 4.0 plus 4.2 plus 4.58 equals 28.78. Equals 3.5875. Mean equals 8. 2.3.0 plus 3.2 plus 3.5 plus 3.8 plus 4.0 plus 4.2 plus 4.5 equals 8. 28.7 equals 3.5875. Step 2. Calculate the standard deviation. Variance equals coefficient of variation, CV, is a useful metric for comparing the relative variability of traits, populations, and ecological variables. It allows researchers to normalize data across different units, aiding in cross-study comparisons in the interpretation of ecological or evolutionary dynamics. You said, correlation. ChatGPT said, ChatGPT. Topic, correlation in ecology and evolution. Recall. Correlation is a statistical measure that describes the degree to which two variables move in relation to each other. It indicates whether and how strongly pairs of variables are related. In ecology and evolution, understanding the correlation between variables can provide insights into patterns and relationships within ecosystems, populations, or species traits. The correlation coefficient, usually denoted as R, R ranges from -1 to 1. R equals 1. R equals 1. Perfect positive correlation. As one variable increases, the other also increases in a perfectly linear fashion. R equals -1. R equals -1. Perfect negative correlation. As one variable increases, the other decreases in a perfectly linear fashion. R equals 0. R equals 0. No correlation. There is no predictable relationship between the variables. The most common form of correlation is the Pearson correlation, which assumes a linear relationship between two continuous variables. Formula for Pearson correlation coefficient. Correlation helps in understanding the relationship between two variables. In ecological and evolutionary studies, the correlation coefficient can be used to describe ecological instance. The correlation between two species population sizes can indicate whether they are positively related, e.g., through mutualistic interactions, or negatively related, e.g., competition. Relationships. In evolutionary biology, correlation is used to determine if two traits, e.g., beak size and body mass in birds, are related. A positive correlation would mean that larger birds tend to have larger beaks, whereas a negative correlation might suggest that larger birds have smaller beaks due to evolutionary constraints or ecological factors. Positive correlation. In ecology, a positive correlation could be seen between temperature and plant growth in a region, where warmer temperatures lead to increased plant productivity. In evolution, a positive correlation could occur between genetic diversity and reproductive success, where higher genetic diversity within a population might be linked to greater reproductive fitness. Negative correlation. In ecology, negative correlations might be found between predator abundance and prey abundance, where an increase in predator numbers leads to a decrease in prey populations. In evolution, a negative correlation could arise between clutch size and egg size, where larger eggs result in fewer eggs being laid due to energy constraints. Zero or no correlation. In some cases, two variables may not be related at all, such as the color of a plant and the density of soil particles in its habitat, suggesting no correlation between these two factors. See application. Correlation is frequently used in ecological and evolutionary research to investigate relationships between variables and to test hypotheses. Correlation is frequently used in ecological and evolutionary research to investigate relationships between variables and to test hypotheses. Ecological applications. Population dynamics. Correlation can be used to understand how environmental variables, like temperature or rainfall, are related to species population growth. For example, researchers might examine the correlation between precipitation levels and plant productivity in arid regions. Species interactions. Ecologists may study the correlation between the populations of different species, e.g., predator and prey, to determine how changes in one species population affect the other. Evolutionary applications. Trait correlations. In evolutionary biology, the correlation between different traits can reveal important evolutionary strategies. For example, a study may show a positive correlation between body size and lifespan in mammals, suggesting that larger mammals tend to live longer due to fewer predators or slower metabolism. Genetic correlations. Correlation can also be applied to genetic traits. A genetic correlation between two traits suggests that the same genes or linked genes are influencing both traits. This is important in studies of evolutionary trade-offs, such as between fecundity and offspring size. Conservation biology. In conservation biology, correlation can be used to understand the relationship between various environmental and biological factors. For example, the correlation between habitat loss and species extinction can help identify critical thresholds for conservation efforts. D. Analysis and synthesis. In ecological and evolutionary contexts, interpreting correlation requires careful analysis to ensure that the relationship between variables is not spurious. While a high correlation might suggest a relationship, it does not imply causation. For example, a positive correlation between species diversity and habitat complexity does not necessarily mean that increased habitat complexity directly causes higher species diversity. Other factors, e.g., climate, resource availability, might be driving both variables. Spurious correlations can also arise when two variables seem related due to a third factor confounder. First, there might be a strong correlation between the number of ice cream sales and the number of shark attacks in a coastal area, but the real underlying factor could be seasonality. Both ice cream sales and shark attacks increase in the summer. When using correlation in research. Causality versus correlation. Careful analysis is needed to differentiate between causal relationships and correlational relationships. For instance, a study might show a strong correlation between rainfall and vegetation growth, but rainfall does not directly cause vegetation growth without considering other variables like soil nutrients. Ecological and evolutionary patterns. Understanding correlations helps researchers recognize patterns in ecosystems and evolutionary processes. For example, a negative correlation between population size and genetic diversity in an isolated population can indicate that the population is undergoing genetic drift due to its small size. Statistical considerations. Correlation analysis requires data that meet assumptions such as normality, linearity, and homoscedasticity. Violating these assumptions might lead to misleading interpretations. Researchers often use Spearman's rank correlation when data do not meet Pearson's assumptions or when the relationship is nonlinear. Practice questions. Previous GATE papers. Q1. A study found a correlation coefficient of +0.75 between body size and reproductive output in a population of fish. What does this imply about the relationship between these two variables? Answer. A correlation coefficient of +0.75 indicates a strong positive correlation between body size and reproductive output. It suggests that as body size increases, reproductive output also increases, implying that larger individuals in the fish population tend to have higher reproductive success. Q2. Following data on plant height X and flower number Y for 5 plants. Plant height X, flower number Y 11520. 28253203042. Researcher observes a negative correlation between temperature and metabolic rate in a species of amphibians. What would this suggest about their physiological response to temperature? A researcher observes a negative correlation between temperature and metabolic rate in a species of amphibians. What would this suggest about their physiological response to temperature? Answer. A negative correlation between temperature and metabolic rate suggests that as the temperature increases, the metabolic rate decreases. This can indicate that the species has a thermoregulatory mechanism where it lowers its metabolic rate in warmer temperatures, possibly to conserve energy or avoid overheating, which is common in ectothermic animals like amphibians. Conclusion. Correlation is a key tool in understanding relationships between variables in ecological and evolutionary research. Insights into how different factors influence each other and allows researchers to formulate hypotheses and interpret complex biological and ecological patterns. However, it is essential to remember that correlation does not imply causation, and careful analysis is needed to draw valid conclusions. You said, ST. ChatGPT said, ChatGPT. Topic, standard deviation ST in ecology and evolution, recall. The standard deviation ST is a statistical measure that quantifies the amount of variation or dispersion of a set of values. In ecology and evolution, the standard deviation is used to describe the spread of data points in a distribution, helping to understand the diversity or variability within a population or an ecosystem. Low ST indicates that the values tend to be close to the mean, implying low variability. High ST indicates that the values are spread out over a larger range, suggesting high variability. Formula for standard deviation ST for a population. The standard deviation ST is a statistical measure that quantifies the amount of variation or dispersion of a set of values. In ecology and evolution, the standard deviation is used to describe the spread of data points in a distribution, helping to understand the diversity or variability within a population or an ecosystem. Low ST indicates that the values tend to be close to the mean, implying low variability. High ST indicates that the values are spread out over a larger range, suggesting high variability. Formula for standard deviation ST for a population. The standard deviation is given by ST equals 1 n area summation I equals 1 n xi minus mu 2 ST equals n1. I equals 1 n area summation n xi minus mu 2, where xi is each individual data point, mu. Mu is the mean of the population, n is the total number of data points in the population. For a sample, the formula is adjusted slightly by using n minus 1. N minus 1 instead of n in the denominator to correct for bias. Sample ST equals 1 n minus 1 n area summation I equals 1. Standard deviation is essential for understanding the distribution of data in ecological and evolutionary contexts. Ecological context. Abundance. Ecological study. If the population size of a species is measured in different regions, the standard deviation can help assess whether a species is uniformly distributed or if there is significant variability in its distribution. Environmental variables. In climate studies, the ST can be used to measure the variability in temperature, rainfall, or other environmental factors, which in turn affect the species distribution in ecological processes. Evolutionary context. Phenotypic variation. In evolutionary biology, ST is used to measure the variation in traits such as size, color, or fitness within a population. High variation might indicate the potential for natural selection to act upon different traits, while low variation could indicate stabilizing selection. Genetic variation. Standard deviation also measures genetic variability, which is critical for understanding the potential for evolution in a population. Populations with low genetic variability, low ST and genetic traits, may be more vulnerable to extinction, while populations with higher ST might have greater evolutionary potential. See application. Standard deviation is applied in many ecological and evolutionary studies. Ecological studies. Habitat fragmentation. When studying how fragmentation of a habitat affects species distribution, researchers may calculate the ST of species density across various fragments. A high ST would indicate that the density varies significantly across patches, suggesting that some areas may be more suitable for species than others. Population studies. In population ecology, ST can be used to measure the stability of a species population over time. For example, if the population size of a species fluctuates significantly from year to year, the ST of population sizes would be high, indicating instability. Evolutionary studies. Evolution. Standard deviation helps to quantify how traits evolve across generations. The ST of a trait like body size is large. It suggests that there is strong phenotypic diversity in the population, possibly due to different selective pressures or genetic variation. If different populations of a species are exposed to different environmental pressures, e.g., temperature, researchers may use ST to compare how these pressures shape the variability in traits like temperature tolerance. A large ST in one population could suggest that a wider range of tolerance exists within that group. Conservation biology. In conservation studies, ST is used to assess the health of populations and ecosystems. Genetic diversity. A population with a low ST in its genetic traits may be at risk of inbreeding depression, which could hinder its long-term survival and ability to adapt to environmental changes. Habitat quality. In evaluating the quality of a habitat, the ST of species richness across different regions or sites can indicate how uniformly or variably species are distributed, providing insights into the ecological integrity of the habitat. D. Analysis and synthesis. While the ST provides valuable insights into variability, its interpretation in ecology and evolution must be done cautiously. Ecological data. In ecological studies, ST can sometimes be misleading if the data is skewed or contains outliers. For instance, in a study on species population densities, a small group of areas with very high densities may result in a high ST, even if most areas have very low densities. In such cases, other statistical measures such as the coefficient of variation, CV, might be more useful, as it normalizes the ST relative to the mean. Evolutionary data. In evolutionary studies, SC helps quantify how variable traits are within a population. However, SC alone does not provide a complete understanding of the evolutionary processes. It should be used in combination with other tools like histograms, regression analysis, and analysis of variance ANOVA to gain deeper insights into how natural selection, genetic drift, or gene flow might be shaping the population. For example, while SC can show high variability in a trait, it does not tell us if the variation is beneficial, adaptive, or neutral. Further analysis would be needed to connect SC with fitness outcomes or evolutionary trends. Data assumptions. The SC assumes that the data follows a normal distribution. If the data is not normally distributed, which is often the case in ecological and evolutionary data, the interpretation of the SC can be misleading. In such cases, non-parametric measures like the interquartile range, IQR, may be more appropriate. Practice questions. Previous gate papers. Q1. In a population of plants, the mean height is 30 centimeters, and the standard deviation is 5 centimeters. What percentage of the plants are expected to have heights between 25 centimeters and 35 centimeters, assuming a normal distribution? Answer. In a normal distribution, 68% of the values fall within one standard deviation of the mean. Therefore, the percentage of plants with heights between 25 centimeters, 30 to 5, and 35 centimeters, 30 plus 5, is approximately 68%. Q2. In an ecological study, the number of individuals in a species across 5 different habitats is given as 10. Standard deviation is a critical tool for measuring variability in both ecological and evolutionary data. It helps researchers understand the diversity and distribution of traits, populations, or environmental variables. However, it must be interpreted carefully, especially in non-normally distributed data, and used in conjunction with other statistical tools for a more comprehensive understanding of ecological and evolutionary patterns. You said, correlation, etc. ChatGPT said, ChatGPT. Topic, correlation in ecology and evolution, recall. Correlation is a statistical measure that expresses the degree to which two variables are related. In ecology and evolution, understanding the relationship between different variables, e.g., species abundance, environmental factors, or genetic traits, is essential to interpret ecological patterns, evolutionary trends, and ecological interactions. Positive correlation. When two variables increase together, e.g., as temperature increases, species richness might also increase. Negative correlation. When one variable increases while the other decreases, e.g., as elevation increases, species diversity might decrease. Zero or no correlation. When there is no predictable relationship between the two variables, e.g., no correlation between species abundance and soil pH in a particular study area. Pearson's correlation coefficient R. Pearson's R is the most common measure of linear correlation between two variables and is defined as R equals n array summation xi minus mu x yi minus mu y n array summation xi minus mu x 2 n array summation yi minus mu y 2 R equals n array summation xi minus mu x 2 n array summation yi minus mu y 2 n array summation xi minus mu x yi minus mu y, where species abundance and environmental factors. Researchers may measure how species abundance correlates with environmental factors such as temperature, rainfall, or habitat complexity. A positive correlation might suggest that higher rainfall leads to greater plant or animal abundance. Resource availability and growth rates. In plant ecology, the correlation between nutrient availability, like nitrogen levels, and plant growth is commonly studied. A positive correlation may show that plants grow faster in soils with higher nitrogen content. Evolutionary context. Genetic traits and fitness. In evolutionary biology, correlation is often used to explore the relationship between genetic traits, like allele frequencies, and fitness. For example, a positive correlation between a particular gene and survival in a given environment may suggest that gene is advantageous for survival and may be subject to natural selection. Correlations. Evolutionary biologists may also study correlations between different traits, such as body size and reproductive success, or beak size and food availability in birds. A strong positive correlation could indicate that the traits are linked through evolutionary processes. See application. Ecological applications. Species-environment correlations. In community ecology, researchers often use correlation to examine how species distributions correlate with environmental gradients, e.g., temperature, pH, or moisture. For example, a positive correlation between plant species richness and soil nitrogen levels could suggest that nutrient-rich soils support greater plant diversity. Population dynamics. In population ecology, the relationship between the population size of two different species might be explored. A positive correlation between two species population sizes could suggest mutualism or facilitation, while a negative correlation could indicate competition or predation. Evolutionary applications. Adaptive evolution. Evolutionary studies often use correlation to assess how phenotypic traits, such as body size or coloration, correlate with fitness, e.g., survival or reproductive success. Positive correlations could indicate that these traits are under positive selection. Genetic correlation. In evolutionary genetics, correlation helps to understand how traits that are genetically linked may evolve together. For example, a strong positive genetic correlation between body size and fecundity might suggest that both traits are evolving together under natural selection. Conservation biology. Habitat and species richness. Conservationists often use correlation to determine how the quality of a habitat correlates with species diversity. A positive correlation between habitat quality and species richness suggests that conserving high-quality habitats is crucial for biodiversity preservation. Climate change studies. In the context of climate change, correlation can help predict how species or ecosystems may respond to changing environmental conditions. A strong positive correlation between temperature increases and the spread of certain invasive species could inform climate change adaptation strategies. D. Analysis and synthesis. Correlation provides valuable insights, but it is important to interpret it carefully. Causality versus correlation. Correlation does not imply causation. For instance, while a positive correlation between forest cover and bird diversity exists, it does not mean that forest cover directly causes bird diversity. Other factors, such as habitat quality or predation pressures, may also be involved. Spurious correlation. Sometimes, two variables may appear to be correlated due to an external factor. For example, an apparent correlation between ice cream sales and drowning rates in summer is spurious because both are affected by warmer temperatures, but one does not cause the other. Limitations in ecological and evolutionary studies. Nonlinear relationships. Pearson's correlation measures only linear relationships. If the relationship between the variables is nonlinear, then other methods like Spearman's rank correlation or regression analysis may be more appropriate. Data quality. Correlation analysis assumes that the data are of good quality and free from errors or outliers. In ecological and evolutionary data, particularly with field studies, this assumption may not always hold. Temporal and spatial scales. Correlation coefficients may vary depending on the temporal and spatial scales of data collection. For instance, species interactions may show a strong correlation over short periods but no correlation over longer time frames. Practice questions. Previous GATE papers. Q1. In a study, the species richness of plants is measured in different plots with varying soil nitrogen levels. The following data is collected. Soil nitrogen level, 0.2, 0.5, 0.7, 1.0, 1.3, species richness, 3, 5, 6, 7, 8. Calculate the correlation coefficient between soil nitrogen levels and species richness. Answer. Using Pearson's correlation formula, the calculations are as follows. Mean of soil nitrogen, mu. X equals 0.2 plus 0.5 plus 0.7 plus 1.0 plus 1.35 equals 0.74 mu. X equals 5. 0.2 plus 0.5 plus 0.7 plus 1.0 plus 1.3 equals 0.74. Mean of species richness, mu. Y equals 3 plus 5 plus 6 plus 7 plus 8, 5 equals 5.8 mu. Y equals 5. 3 plus 5 plus 6 plus 7 plus 8 equals 5.8. Next, calculate the covariance and then the correlation coefficient R. R. The positive value of R. R will indicate a positive correlation between nitrogen levels and species richness. Q2. In an evolutionary study, the following data represents the relationship between the number of offspring produced and parental body size for two different species. Offspring X2, 3, 4, 5, 6 body size Y 45, 50, 52, 55, 60. What can be inferred about the relationship between body size and offspring number? Answer. Since the correlation coefficient calculated using Pearson's formula is likely to be positive, this suggests that larger body size may be associated with more offspring, implying a positive correlation between body size and reproductive success in the species. Q3. A study finds a negative correlation between the availability of food resources and the population of a predator species. What does this imply about the predator-prey relationship? Answer. A negative correlation indicates that as food resources for the predator decrease, the predator population also declines. This could suggest that the predator is highly dependent on the availability of prey food, and a reduction in prey leads to a reduction in predator numbers. This may be consistent with bottom-up regulation in predator-prey dynamics. Conclusion. Correlation is a vital tool in understanding the relationships between variables in both ecological and evolutionary studies. Whether studying the effects of environmental factors on species distribution or examining how genetic traits influence fitness, correlation allows researchers to draw connections and make predictions. However, it is essential to remember that correlation alone does not confirm causality and that careful analysis is necessary to avoid misleading conclusions. You said. What is hypothesis testing? Why? What if don't do it? Common mistakes ChatGPT said. ChatGPT. Hypothesis testing. Recall. Hypothesis testing is a statistical method used to assess whether there is enough evidence in a sample of data to support or reject a specific hypothesis about a population. It involves comparing observed data with a null hypothesis, usually representing no effect or no relationship, and determining the likelihood that the data would occur if the null hypothesis were true. Steps in hypothesis testing. Formulation of hypotheses. Null hypothesis H. This hypothesis suggests that there is no effect, difference, or relationship. It is the hypothesis that is tested. Alternative hypothesis H or HA. This is the hypothesis that indicates the presence of an effect, difference, or relationship. Choose a significance level alpha. The significance level is the probability of rejecting the null hypothesis when it is actually true. Type 1 error. Common values are 0.05 or 0.01. Test statistic. A test statistic e.g., t statistic, z statistic is calculated from a sample data. It is used to determine how much the sample data deviates from the null hypothesis. P-value. The p-value represents the probability of obtaining the observed sample data, or something more extreme, given that the null hypothesis is true. If the p-value is smaller than the significance level alpha, we reject the null hypothesis. Conclusion. Based on the p-value and significance level, a conclusion is drawn. If p is less than or equal to alpha, reject the null hypothesis. Evidence supports the alternative hypothesis. If p and gt, alpha, fail to reject the null hypothesis, no sufficient evidence to support the alternative hypothesis. B. Why hypothesis testing is important. Objective decision making. Hypothesis testing allows researchers to make objective decisions based on data. Without it, conclusions could be based on personal biases, leading to false interpretations. Scientific method. Hypothesis testing is a core part of the scientific method. It allows researchers to validate or refute hypotheses with empirical evidence, thus advancing knowledge in various fields, including ecology, evolution, medicine, and social sciences. Evidence-driven conclusions. Hypothesis testing ensures that conclusions about population parameters are based on evidence rather than assumptions. This is critical in scientific research, where evidence-based findings are needed for reliability and credibility. Risk management. It helps in quantifying the risk of making incorrect conclusions. By setting a threshold alpha, researchers can control the probability of making a type 1 error. See what happens if you don't perform hypothesis testing. Inaccurate conclusions. Without hypothesis testing, decisions and conclusions may be drawn from personal bias, anecdotal evidence, or random chance. This increases the risk of drawing incorrect conclusions and publishing unreliable findings. Overlooking statistical significance. In scientific studies, not conducting hypothesis testing might lead to missing out on important patterns or relationships that could have been identified with proper statistical testing. For instance, you may fail to detect a true effect or relationship because you didn't formally assess its significance. Risk of type 1 and type 2 errors. Type 1 error, false positive, concluding that there is an effect when there is none, rejecting a true null hypothesis. Type 2 error, false negative, concluding that there is no effect when there actually is one, failing to reject a false null hypothesis. Both errors can have serious consequences, especially in fields like medicine or environmental science. Lack of rigor in research. Without hypothesis testing, research may lack scientific rigor. Hypothesis testing helps validate claims and ensures that findings are based on statistical evidence, thus improving the reliability and validity of research conclusions. D. Common mistakes in hypothesis testing. Misinterpreting p-values. One common mistake is misinterpreting the p-value. A p-value smaller than 0.05 does not mean that the null hypothesis is false. It simply means there is strong evidence against the null hypothesis. A p-value above 0.05 doesn't prove the null hypothesis is true, it just suggests insufficient evidence to reject it. Ignoring effect size. While hypothesis testing helps determine if a result is statistically significant, it does not inform you about the size or importance of the effect. Researchers may focus solely on achieving a statistically significant result without considering the practical significance of the findings. Always assess the effect size to understand the magnitude of the relationship or effect. Multiple testing errors. When conducting multiple hypothesis tests, the likelihood of making a type 1 error increases. Researchers often fail to adjust for this risk, leading to false positives. To avoid this, methods such as the Bonferroni correction or false discovery rate (FDR) control should be used when multiple tests are performed. Overlooking assumptions. Hypothesis test. Multiple testing errors. When conducting multiple hypothesis tests, the likelihood of making a type 1 error increases. Researchers often fail to adjust for this risk, leading to false positives. To avoid this, methods such as the Bonferroni correction or false discovery rate (FDR) control should be used when multiple tests are performed. Overlooking assumptions. Hypothesis tests are based on certain assumptions, e.g., normality of data, independence of observations. Failing to check these assumptions can result in misleading conclusions. For example, using a t-test without checking for normality in small sample sizes can lead to inaccurate results. Stop data collection too early. In some cases, researchers may stop collecting data prematurely after obtaining significant results. This is called data peaking. It increases the risk of type 1 errors and reduces the reliability of the findings. It's important to plan the sample size and analysis in advance. Failure to consider power. Power analysis is crucial in hypothesis testing because it estimates the probability of detecting an effect when there's one, i.e., avoiding type 2 errors. Small sample sizes or insufficient power can lead to non-significant results even when an effect exists. Researchers should perform a power analysis before conducting studies to ensure their study is sufficiently powered. Example of hypothesis testing in ecology. Scenario: you are studying the effect of fertilizer on plant growth in two different soil types. You hypothesize that the fertilizer will significantly increase plant height in both soil types. Null hypothesis H. Fertilizer has no effect on plant height, i.e., the mean height with fertilizer is equal to the mean height without fertilizer. Alternative hypothesis H. Fertilizer has an effect on plant height, i.e., the mean height with fertilizer is different from the mean height without fertilizer. By performing hypothesis testing, e.g., t-test, you can assess if the observed difference in plant height is statistically significant or if it could have occurred due to random chance. If the p-value is lower than the significance level, e.g., 0.05, you reject the null hypothesis and conclude that the fertilizer has a significant effect on plant height. Conclusion. Hypothesis testing is a fundamental part of scientific research and provides a structured framework for making decisions based on data. It allows researchers to assess the evidence for or against a hypothesis objectively, helping to prevent biases and errors in research. However, failure to perform hypothesis testing or mistakes in its application can lead to inaccurate conclusions, poor decision-making, and unreliable scientific findings. Therefore, it is crucial to conduct hypothesis testing rigorously and interpret the results cautiously. You said. Statistical hypothesis testing. ChatGPT said. ChatGPT. Statistical hypothesis testing. Statistical hypothesis testing is a method used to make inferences or draw conclusions about a population based on sample data. It is integral to statistics and is employed in various fields, including ecology, evolution, medicine, social sciences, and more. 1. Key concepts in statistical hypothesis testing. Hypothesis. A hypothesis is a testable statement or assumption about a population parameter, e.g., mean, proportion. It is typically framed as two competing hypotheses. Null hypothesis H, the default assumption, usually stating that there is no effect, difference, or relationship between variables. For example, there is no difference in species diversity between two habitats. Alternative hypothesis H or HA, the hypothesis that contradicts the null hypothesis, suggesting that there is an effect, difference, or relationship. For example, there is a difference in species diversity between two habitats. 2. Steps in statistical hypothesis testing. State the hypotheses. Null hypothesis H, the assumption that there is no effect or relationship. Alternative hypothesis H, the hypothesis that there is an effect or relationship. Example, in studying the effect of climate change on plant species, the null hypothesis could be, climate change has no effect on plant species distribution. The alternative hypothesis could be, climate change affects plant species distribution. Choose a significance level alpha. The significance level alpha is the threshold for determining whether to reject the null hypothesis. Common values are, alpha equals 0.05, 5% level of significance. Alpha equals 0.01, 1% level of significance. This represents the probability of making a type 1 error, rejecting a true null hypothesis. Select the appropriate test. Depending on the type of data, e.g., continuous, categorical, the sample size, and whether the population standard deviation is known, different statistical tests are applied. Common tests include, t-test for comparing means of two groups. ANOVA for comparing means across multiple groups, chi-square test for categorical data, regression analysis for testing relationships between variables, calculate the test statistic. A test statistic is calculated based on sample data. It measures how far the sample statistic is from the population parameter under the null hypothesis. Example, for a t-test, the test statistic t is calculated by the formula t equals sample mean minus population mean sample standard deviation n t equals sample standard deviation n sample sample mean minus population mean, where sample mean equals average of the sample data, population mean equals the expected population value under the null hypothesis, sample standard deviation equals the standard deviation of the sample, n equals sample size. Compute the p-value. The p-value is the probability of observing the sample data, or something more extreme, under the assumption that the null hypothesis is true. If p is less than or equal to alpha, you reject the null hypothesis, indicating that the sample data provides enough evidence to support the alternative hypothesis. If p and gt, alpha, you fail to reject the null hypothesis, suggesting that the evidence is insufficient to support the alternative hypothesis. Make a decision. Based on the p-value in the chosen significance level alpha, you make a decision. Reject h, if the p-value is less than or equal to the significance level p is less than or equal to alpha. Fail to reject h, if the p-value is greater than the significance level p and gt, alpha. Draw a conclusion. If the null hypothesis is rejected, conclude that there is significant evidence in favor of the alternative hypothesis. If the null hypothesis is not rejected, conclude that there is insufficient evidence to support the alternative hypothesis. 3. Types of errors in hypothesis testing. Type 1 error, false positive, occurs when the null hypothesis is rejected when it is actually true. This error is denoted by the probability alpha significance level. For example, concluding that climate change significantly affects plant distribution when it actually does not. Type 2 error, false negative, occurs when the null hypothesis is not rejected when it is actually false. This error is denoted by beta. For example, failing to detect an effect of climate change on plant distribution when such an effect actually exists. 4. Statistical power. Power is the probability of correctly rejecting the null hypothesis when it is false, i.e., detecting an effect when there is one. It is influenced by sample size, and larger sample sizes increase power. Effect size. Larger effect sizes, greater differences or stronger relationships increase power. Significance level alpha, higher alpha levels increase power, but also increase the risk of type 1 errors. Variability of data. Less variability in the data increases power. 5. Common statistical tests in hypothesis testing. 1 sample t-test, used to test whether the mean of a single sample differs from a known value, e.g., population mean. Example, does the average height of a species of plant differ from a known population value? 2 sample t-test, used to compare the means of 2 independent groups. Example, is there a difference in plant growth between 2 different habitats? Paired t-test, used when comparing the means of 2 related groups, e.g., before and after treatment. Analysis of variance, ANOVA, used when comparing the means of more than 2 groups. Example, does plant species diversity vary across different environmental conditions, e.g., dry, wet, and temperate? Five. Common statistical tests in hypothesis testing. 1 sample t-test, used to test whether the mean of a single sample differs from a known value, e.g., population mean. Example, does the average height of a species of plant differ from a known population value? 2 sample t-test, used to compare the means. 1 sample t-test, used to test whether the mean of a single sample differs from a known value, e.g., population mean. 1 sample t-test, used to test whether the mean of a single sample differs from a known value, e.g., population mean. Example, does the average height of a species of plant differ from a known population value? 2 sample t-test, used to compare the means of 2 independent groups. Example, is there a difference in plant growth between 2 different habitats? Paired t-test, used when comparing the means of 2 related groups, e.g., before and after treatment. Analysis of variance, ANOVA, used when comparing the means of more than 2 groups. Example, does plant species diversity vary across different environmental conditions, e.g., dry, wet, and temperate? Chi-square test, used for categorical data to test the relationship between 2 variables, e.g., the association between 2 species presence in different habitats. 6. Why hypothesis testing is crucial in research. Objective decision making. Hypothesis testing provides an objective framework for making decisions based on data, reducing the influence of researcher biases. Statistical significance. It allows researchers to determine whether observed differences or relationships in data are statistically significant or likely due to random chance. Scientific rigor. Hypothesis testing ensures that scientific findings are based on systematic, reproducible evidence rather than anecdotal or speculative conclusions. Control errors. It helps researchers control type 1 and type 2 errors, improving the accuracy and reliability of conclusions. 7. Common mistakes in hypothesis testing. Misinterpreting p-values. A common mistake is to equate a low p-value, e.g., p-NLT, 0.05 with the null hypothesis being false. A p-value only tells you the probability of observing the data if the null hypothesis is true. It doesn't prove that the null hypothesis is false. Not checking assumptions. Many tests like t-tests and ANOVA have assumptions, e.g., normality, independence, homogeneity of variances. Failing to check these assumptions can lead to incorrect conclusions. Overlooking effect size. Focusing only on whether a result is statistically significant, p-value, without considering the magnitude of the effect size can be misleading. A small effect with a large sample size can be statistically significant but not practically meaningful. Multiple comparisons without correction. When performing multiple hypothesis tests, the likelihood of a type 1 error increases. Researchers should apply corrections, e.g., Bonferroni correction, when making multiple comparisons. Example scenario in ecology. Hypothesis. Researchers want to test whether the introduction of a new invasive species affects the population size of native plants. Null hypothesis H. The introduction of the invasive species does not affect the population size of native plants. Alternative hypothesis H. The introduction of the invasive species affects the population size of native plants. After collecting data on plant populations before and after the introduction of the invasive species, the researchers use a paired t-test to analyze the data and calculate a p-value. If p is less than or equal to alpha, the null hypothesis is rejected, and the researchers conclude that the invasive species has a significant effect on native plant populations. If p and gt, alpha, the null hypothesis is not rejected, suggesting no significant effect. Conclusion. Statistical hypothesis testing is a powerful tool in research that allows scientists to draw objective conclusions based on sample data. By following a structured process of hypothesis formulation, test selection, and decision-making, researchers can assess the significance of their findings and control for errors. However, careful attention must be given to common mistakes such as misinterpreting p-values, overlooking assumptions, and failing to consider effect sizes to ensure reliable and accurate conclusions. You said, concept of p-value. ChatGPT said, ChatGPT. Concept of p-value. The p-value is a fundamental concept in statistical hypothesis testing. It provides a measure of the strength of evidence against the null hypothesis. In simpler terms, the p-value helps determine whether the observed data is consistent with the null hypothesis or if it is sufficiently unusual to reject the null hypothesis in favor of the alternative hypothesis. 1. What is a p-value? The p-value represents the probability of obtaining an observed result, or one that is more extreme, given that the null hypothesis is true. It is a measure of the compatibility of the sample data with the null hypothesis. P-value typically is less than or equal to 0.05 indicates that the observed data is unlikely to have occurred if the null hypothesis were true, providing evidence in favor of rejecting the null hypothesis. A large p-value, typically in GT, 0.05, suggests that the observed data is consistent with the null hypothesis, and there is insufficient evidence to reject it. 2. How is the p-value interpreted? Rejecting the null hypothesis. If the p-value is less than or equal to the chosen significance level alpha, typically 0.05, the result is statistically significant, and the null hypothesis is rejected. This means the observed effector relationship is unlikely to be due to random chance alone. Example, if p equals 0.03 and alpha equals 0.05, we reject the null hypothesis, concluding that the effect observed, e.g., a change in species diversity, is significant. Failing to reject the null hypothesis. If the p-value is greater than the significance level alpha, the result is not statistically significant, and we fail to reject the null hypothesis. This means that the evidence is insufficient to support the alternative hypothesis, and the observed effect may be due to random variation. Example, if p equals 0.08 and alpha equals 0.05, we fail to reject the null hypothesis, suggesting that there is no significant evidence to support the alternative hypothesis. 3. P-value in the context of hypothesis testing. Null hypothesis H, there is no effect, difference, or relationship in the population. Alternative hypothesis H, there is an effect, difference, or relationship in the population. The p-value tells you how likely it is to observe your data, or more extreme data, under the assumption that the null hypothesis is true. For example, in a study investigating the effect of a new drug, the null hypothesis might be that the drug has no effect on the disease. The p-value will tell you whether the observed improvement in patient outcomes is unlikely to have occurred by chance. 4. The role of significance level alpha. The significance level alpha is a threshold chosen by the researcher to determine the cutoff for rejecting the null hypothesis. Commonly used values are 0.05, 0.01, and 0.10. If p is less than or equal to alpha, reject the null hypothesis. If p and GT, alpha, fail to reject the null hypothesis. For example, if alpha equals 0.05 and p equals 0.03, you reject the null hypothesis. If alpha equals 0.05 and p equals 0.07, you fail to reject the null hypothesis. 5. Misconceptions about the p-value. The p-value does not measure the probability that the null hypothesis is true. The p-value measures the probability of the observed data, or something more extreme, assuming the null hypothesis is true. It does not tell you the probability of the null hypothesis itself being true or false. A smaller p-value does not always imply a more important result. A p-value less than 0.05 does not necessarily mean that the observed effect is practically significant or important. It only indicates that the observed effect is statistically significant, but the effect size should be considered as well. The p-value is not the same as the probability of making a type 1 error. The p-value is not the probability of making a type 1 error, incorrectly rejecting a true null hypothesis. Instead, it is the probability of observing the data if the null hypothesis is true. 6. Example of p-value in ecology. Imagine you are conducting an experiment to test whether a particular fertilizer improves the growth of a plant species in a specific habitat. Your hypotheses are: null hypothesis H, fertilizer has no effect on plant growth, mean growth with fertilizer is equal to mean growth without fertilizer. Alternative hypothesis H, fertilizer has an effect on plant growth, mean growth with fertilizer is different from mean growth without fertilizer. After performing a statistical test, e.g., t-test, you obtain a p-value of 0.03. If your significance level alpha is 0.05, since p is less than or equal to alpha, you reject the null hypothesis. This suggests that the fertilizer likely has a significant effect on plant growth. However, the effect size magnitude of the fertilizer's effect should also be considered to understand the practical significance of the result. 7. The role of p-value in scientific research. Objective decision-making. The p-value helps researchers make objective decisions about whether to reject the null hypothesis based on data. This reduces the influence of bias or subjective judgment. Guiding further research. A statistically significant p-value may indicate a need for further studies to explore the effect in more detail, refine the research question, or test the findings in different contexts. Reliability of findings. Researchers rely on p-values to report the statistical significance of their findings, which is crucial for the credibility and reproducibility of scientific research. 8. Limitations of the p-value. P-hacking. Data dredging. P-hacking refers to manipulating data or testing multiple hypotheses to achieve a desired p-value, usually less than 0.05. This can lead to false positives and unreliable conclusions. Over-reliance on p-values. Solely focusing on whether the p-value is below the significance level can lead to misinterpretation. It is essential to also consider other factors, such as the effect size, confidence intervals, and the context of the study. P-value is a dichotomy. The idea of significant versus not significant, based purely on a p-value threshold, like 0.05, is criticized for oversimplifying the complexity of scientific findings. This dichotomy does not capture the uncertainty in scientific data. Does not measure the size of an effect. A small p-value indicates that the result is unlikely under the null hypothesis, but it does not tell you the magnitude of the effect. Thus, a result may be statistically significant but not necessarily practically important. Conclusion. The p-value is an essential tool in hypothesis testing, offering a measure of how well the sample data aligns with the null hypothesis. It guides researchers in making decisions about rejecting or failing to reject the null hypothesis. However, it is important to interpret p-values cautiously and in context, considering factors such as effect size, the research design, and potential biases. Understanding the p-value's limitations helps prevent misuse and ensures more robust, credible conclusions in scientific research. You said, type 1 error. ChatGPT said, ChatGPT type 1 error. A type 1 error is a statistical error that occurs when the null hypothesis H is rejected when it is actually true. In other words, it is the incorrect decision to conclude that there is an effect, relationship, or difference in the population, when in reality, there is none. This is also referred to as a false positive. 1. Understanding type 1 error. In hypothesis testing, the null hypothesis H generally represents the status quo, suggesting no effect or relationship between variables. The alternative hypothesis H posits that there is an effect, difference, or relationship. Type 1 error, incorrectly rejecting the null hypothesis when it is true. Example, a researcher claims that a new drug is effective, but in reality, the drug has no effect. The null hypothesis is true. 2. Significance level alpha and type 1 error. The probability of making a type 1 error is denoted by alpha, also called the significance level. The significance level is chosen by the researcher before conducting the hypothesis test. Common values for alpha are 0.05, 5%, or 0.01, 1%. A significance level of alpha equals 0.05 implies that there is a 5% chance of committing a type 1 error, rejecting a true null hypothesis. The lower the significance level, e.g., alpha equals 0.01,